Welcome everyone to the fourth webinar of Obesity 2020, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. Between now and December, we have a number of webinars lined up, all focused on the science being conducted by leading obesity researchers around the world. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This webinar is titled Heterogeneity and Crosstalk of Human Brown Adipose Tissue, and will feature Dr. Camilla Schéele, Associate Professor at the Novo Nordisk Foundation Center for Basic Metabolic Research, and Dr. Christian Wolfrum, Professor in the Department of Health Science and Technology at ETH Zürich. Today, Dr. Schéele and Dr. Wolfrum will discuss the physiology of brown adipose tissue and share late-breaking research on the role of BAT in whole body metabolic regulation. And first, we'd just like to acknowledge our partners at the APS and thank the sponsors of this webinar series for making it all possible. And so without any further ado, I'm very pleased to welcome our first presenter, Dr. Camilla Schéele. Camilla, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Okay, Liam, thank you so much for that introduction, and thanks also to all of you who are listening to this webinar. So today we're going to talk about brown fat. I'm Camilla Schéele, and in the background here you see my workplace in Copenhagen. Everyone knows that in obesity, we get an increase in white adipose tissue, but brown fat activity actually is negatively associated with body mass index. And so brown fat is a special type of adipose tissue that can produce heat in response to cold. And this is an energy consuming process. And what happens is that norepinephrine is released from the nerve ends innervating the adipose tissue, and this results in a lipolysis, so providing the mitochondria with fuel. And but instead of using this for ATP production, the brown fat mitochondria has a special protein called USP1, and this uncouples the mitochondria, and instead heat is released. So in the brown adipocyte, it also, when it's active, it also takes up glucose and lipids from the circulation. So this is why when brown fat is active, we have an increase in energy expenditure, and it also affects the insulin sensitivity, lipid metabolism, and has also an effect on appetite regulation. And some of these effects are also more indirect and can be mediated by factors that are produced and secreted from the brown fat cell, which are called cytokines, which we will talk a little bit more about later in this presentation. So we have much more brown fat as babies. So when we're newborn, we have some brown fat in the back of our in the back, and you can see where the red arrow points to. This depot is actually corresponding to the depot that we usually analyze in mouse models, which is the intrascapular brown fat. Well, as we grow, this depot is lost. So in an adult human, we still have some brown fat, but we don't have the intrascapular brown fat. And we can detect the active brown fat in adult humans by using PET-CT scanning in combination with a glucose tracer. So here I first show you a picture of a person that has been kept warm. And you can see no active brown fat in this individual. But when we take the same person and subject him to cooling for 3 hours, then his brown fat will be active and it will take up glucose. And here you can see a picture of how that looks like. So here you can see, if you compare this picture to the right with a picture from the newborn, you can recognize the depots that are more deep in the body, while you see that the intrascapular brown fat is not there anymore. But we still have some around in the neck area. So there is a variation between individuals on how much brown fat we have, and in some of us, we can't even detect it. There is also a heterogeneity within the brown fat in adults. So as you can see here on this tissue section, there is a mixture of large unilocular adipocytes that have just one lipid droplet, so unilocular, and more multilocular adipocytes, which are positive for USP1 and more represent this brown fat morphology that we're used to. If we go back in time to 1972, there was a study showing, looking at how brown fat morphology was in different age spans. So in children and up to the ages of 20, we can see this homogeneous kind of brown fat. It is almost completely depleted of lipids because it's so active. And then as we grow older, we get more and more lipids, and it becomes also more and more heterogeneous and more similar to white adipose tissue. So the question is, how flexible is this? Can we restore the childhood phenotype of brown fat activity in adults, or does it just go in one direction? So some studies have addressed how much brown fat we can actually have as adults. And one interesting study has been done by Leitner et al. In 2017, where they did a PET CT scanning of 12 individuals and then made an overlay of these PET CT scans. So the red staining that you see represents anywhere where you can find active brown fat in any of these individuals. So then you see that we have, it seems to be much more. And then the blue region represents the fat tissue that is directly connected to this active brown fat. So another model that's interesting to look at is a disease situation which is pheochromocytoma. And here in this disease, we have an overproduction of norepinephrine from the adrenal gland. So this is a tumor in the adrenal gland. And when you have this tumor, we can see that there is an increase in brown fat differentiation or thermogenic fat differentiation in the visceral fat that is nearby the adrenal gland. So you can compare the picture to the left, where a tissue section comes from a healthy person, to the tissue section to the right, where we have more thermogenic kind of cells that are positive for USP1. So here's a case study of a patient with pheochromocytoma, where this person have had this condition for a long time. You can see that there is an extreme amount of brown fat in the classical regions, but also in the visceral abdomen region. And this was done in a room temperature scanning, so he was not even subjected to cold. And after removal of the tumor, this effect was removed as well. So if we compare these conditions in a more cartoon-like way, it's more easy to see the differences. So first we have the normal activation of one individual subjected to cold, and then in the picture in the middle we have the overlay. So this might represent where we actually can get a brown fat activation if we have a full glucose uptake in this fatty tissue. And then the last picture is a little bit different, I would say, because then the brown activation doesn't come from the direct innervation of the adipocytes. It rather is pumped out from adrenaline from the adrenal glands. So it's more of a hormonal activation, which you can also see that the rather vascularized visceral fat has really activated thermogenic differentiation. And this is quite interesting, as it indicates to us that we have progenitor cells that can actually be induced to differentiate into thermogenic adipocytes. So we were quite interested in this fat surrounding the kidneys, the perirenal fat, and we wanted to investigate this further. It has been shown to be some brown adipose tissue there, but we wanted to look into this in more detail. So we recruited healthy kidney donors that were quite not that young. They were in their 60s, and we analyzed the fat surrounding these the kidneys of these donors. We could see that the tissue looked very similar to the brown fat that we find in the supraclavicular deep neck region. It's heterogeneous. It has unilocular lipid fat cells and multilocular fat cells. But when we did a staining, we could see that the unilocular adipocytes also was positive to USP1. So we compare this at a global level with RNA sequencing analysis, and we could see that there was a separation between the subcutaneous adipocytes, which you see here in the top circle, and in the middle circle you have the unilocular perirenal, and then the green samples are the multilocular perirenal. So they were all separating into different groups. And what was also interesting was that there was an accumulation of multilocular adipocytes close by the adrenal gland. So probably due to the higher concentration of norepinephrine coming out of this particular spot. So what we did next was to isolate adipogenic progenitors from all regions surrounding the kidney. And what was very interesting was that regardless of where we took the fat biopsies from for these isolations, we could find thermogenic adipocytes at equal frequency. So this suggested to us that we have a dormant brown fat in the perirenal depot because we have these progenitors still present in all regions, regardless if the mature cells are multilocular or unilocular. We then wanted to look into obesity, what happens here. So we took biopsies from normal weight subjects and obese or overweight subjects, and we compare these using RNA sequencing. And we could see that genes that were involved in cellular respiration or the electron transport chain were downregulated in obesity. And this included, for example, several of the COX genes, which you can see here, the samples from the obese and overweight were more similar to the white adipose tissue. And so we concluded that the thermogenic capacity was downregulated. And this is consistent, of course, with the PET-CT scan studies that show an association with reduced brown fat activity. We also isolated adipogenic progenitors from these subjects, and here we could again see that it didn't depend on where if the person was obese or overweight. We could still find thermogenic adipocytes that were differentiating just fine in vitro. So they responded to norepinephrine in terms of upregulating USP1, and they responded to norepinephrine in terms of increasing their oxygen consumption rate. So we conclude here that the thermogenic capacity is reduced in obesity, but the brown fat identity seems to be kept. And this study is available at bioRxiv. What about the bioarchive? So we know that norepinephrine is a really important player in browning and activating brown fat, but there are also these factors that I mentioned already that are produced and secreted from the brown adipocytes. And here it's also important to know that in the brown fat, there's not only adipocytes, there's also other types of cells. So we have endothelial cells and neural cells and pre-adipocytes, of course, and also immune cells. And all these different cell types actually have an important role in the brown fat maturation or in the thermogenic maturation, because we need a thermogenic differentiation, we need angiogenesis, and we also need an increased innervation of the brown adipose tissue. And there's also some studies showing that the immune cells have an important role in interacting with the thermogenic capacity. There are also studies now showing that cytokines can actually be secreted and interact with metabolism at an organ crosstalk level. So it can interact with the muscle, gut, liver, white fat, and also the brain. So this is a new and very exciting area in terms of obesity research. So we wanted to look more into if we could map some cytokines and maybe identify some new interesting regulators. So we decided to isolate cells from brown and white fat, and we differentiated them in vitro, and we could see that they are actually maintaining their phenotype. So if we isolate cells from brown fat, they also still respond better to norepinephrine in terms of inducing UCP1 compared to the cells isolated from white fat. And this is both in terms of gene expression, but also in terms of oxygen consumption. So these are functional human brown fat cells. And this is a model that we use for several different applications. And one of the studies that we did and published last year was the secretomes of human brown and white adipocytes. And here we were so lucky and happy to have the front page of cell metabolism, and the leaves that you can see here represents the secretomes. So what we did was we isolated cells from white and brown fat from several different individuals, and then we took the cell culture media from these cultures, and we performed proteomics in collaboration with Matthias Mann and Atul Desmukh at the Novo Nordisk Foundation Center for Protein Research. And we found that there were very distinct secretomes between brown and white adipocytes in humans. We found 262 common proteins. Among these, we had a differential regulation of in white and brown fat cells. We found that in the white fat cells, we had a more secretion of extracellular matrix proteins, maybe allowing for more plasticity. And in the brown adipocytes, we had more secretion of complement system factors, suggesting that we have a difference in the innate immune system between white and brown fat cells. And this has also been reported from other studies. So one of the proteins that were only detected in the brown fat media was EPDR1. So we decided to investigate the function of this protein a little bit further. So we used our cell model and performed a knockdown. And we then stimulated the cells with norepinephrine, and we could see that you see the blue bars here. It represents the samples that have been treated with siRNA targeted EPDR1. We don't get the same response in terms of USP1 induction, and the same with the oxygen consumption rate here. The blue line here is the knockdown samples. You can see that it doesn't respond at all as well as the control cells. And we also looked into the knockout mouse, EPDR1 knockout mouse, and here we found that several genes were downregulated that are important for thermogenic identity. And when we did an ex vivo oxygen consumption assay, we could see that there was a less oxygen consumption in the knockout samples. So if we reduce EPDR1 in the precursor cells and differentiate them, or in the knockout mouse, then this will decrease their response to norepinephrine. So to further investigate what's going on here, we did a proteomics analysis following EPDR knockdown in the human cells. And here we could see that GLUT4 was reduced, and also several proteins in the electron transport chain were reduced. So this really explained the phenotype to us. And we conclude that we have a failure in thermogenic differentiation if EPDR1 is not present. We then injected EPDR1 in mice for 3 weeks daily, and then we saw an increase in UCP1 expression in the white adipose, so both in the epididymal and in the inguinal fat was increased. And to investigate if EPDR1 could actually be a circulating factor, even to affect browning of white fat via the vascular network, we measured it in human plasma. And here we couldn't detect it in all samples, but in the samples that we could detect it in, we could see that these subjects had a higher expression of UCP1 in their brown fat. So there's definitely an association between EPDR1 and thermogenic phenotype. So we conclude that EPDR1 mediates thermogenic commitment of pre-adipocytes. So to summarize what I talked about today, we can see that humans have dormant brown fat in adults, and this is in the region of perirenal adipose, in the perirenal adipose region, and we can also see it in obesity, in the supraclavicular brown fat, and this is represented by a downregulation of several thermogenic genes. We can see that there's nothing actually wrong with the adipogenic progenitors. So there seems to be something inhibiting the thermogenic differentiation. And that's why we don't have more thermogenic adipose tissue, we think. And something that can promote this differentiation is different cytokines that can stimulate this. And one of the cytokines that we have identified is EPDR1 that promotes thermogenic differentiation. And with that, I would like to thank my group. So today I talked about data from Naya Jespersen and Loni Pais, who's standing up to the right, and Susanna Söber, who's sitting down to the right. And I would like to thank all our collaborators and our funding bodies. And thank you all for listening this summer day. And with that, I would like to hand over to Christian, who will continue to entertain you on brown fat.
Okay, thank you very much, Camilla. Thanks to the audience for listening. And we heard now a lot about the human adipose tissue, human brown adipose tissue, and how it is formed and which controls its function. And I would like to delve a little bit deeper in the whole plasticity of brown adipose tissue and try to link this a bit to the development of metabolic complications. So just since Camilla introduced everything so very nicely, I can keep this short, but maybe to remind you, since I'm also talking quite a bit about mouse work, we always in the brown adipose tissue field distinguish different types of brown adipocytes or brown-like adipocytes. So we have the classical brown adipose tissue, which is located in the neck region of the mouse, and which you can see on the left side of the slide, which is a very dense tissue with small lipid droplets and lots of mitochondria. And then you obviously know the white adipose tissue, which you find in many different depots over the body. And obviously in obesity, there's this high excess of this tissue. And then you have brown-like adipocytes, which are called either beige or bright, depending on which nomenclature you follow, which develop within normally white adipose tissues. So these are shown here in the middle, and you can see these cells appear upon cold stimulation in rodents in normally white adipocyte depots. I come to this in a second. And in humans, it's still a matter of debate whether humans have mostly beige, bright adipocytes or maybe also some brown adipocytes. And today I'd really like to discuss the plasticity of these cells and thus their possible contribution to energy homeostasis. I talked about the different tissues. Here's just an overview of the mouse. You've seen something before, but I'd still like to show it again because I will talking about these different tissues. You have marked in black the intrascapular brown adipose tissue, which is the classical brown adipose tissue depot with these very dense cells. Then you have white adipose tissue depots like the epididymal or the abdominal adipose tissue in mice, which rarely browns. So no matter how strongly you stimulate with the cold, you get very few brown adipocytes in such, or beige adipocytes in such a depot. And lastly, you have the inguinal adipose tissue or the subcutaneous adipose tissue. In there, you have white adipocytes as well as bright beige adipocytes forming in response to cold stimulus. So we've seen now for the human that the process of differentiation might be not regulated under these conditions, but that the browning might be regulated. And as I promised you, we'll delve a little bit deeper into the whole issue of plasticity. So I want to start off with an experiment that we did already quite a while ago, but I think it's still quite interesting as it illustrates the capacity of adipose tissue to remodel. And this is shown here. So here you have an overview of a white adipose tissue, the inguinal white adipose tissue, in mice that were subjected to cold. So you see here the paradigm that was used. Mice were cooled for 1 week to 8 degrees, and then they were put back to room temperature, which itself is a mild cold stress for mice, one should mention here. And if you can see, the white adipose tissue before stimulation, week 0, looks mostly like you would expect a classical white tissue to look, right? Large unilocular adipocytes, a single lipid droplet, very few dense staining that would indicate the presence of mitochondria. If you now stimulate with cold, you see what you also can observe in humans, the appearance of these beige or bright adipocytes, which contain UCP1, and quite importantly, which have these multilocular structure, which you can probably see better if you zoom in or if I would show this on a big screen. And these denote, or these are what we would consider bright or beige adipocytes. So if we want to understand the plasticity of formation and also conversion, we need to employ certain tracing models. And this is not that trivial in animals because there's no specific markers for specifically beige or brown adipocytes, or especially white adipocytes. So it's not that easy to trace them. But what we can look at is the kinetics, how these cells, which appear upon a cold stimulus, how they disappear when the cold stimulus is removed. And you can see this in the next slide, or in the next graph here. If you remove the stimulus and put mice back to a room temperature, you see after 2 weeks, you still have quite a large number of these multilocular cells. After 3 weeks, it's notably less. And after 6 weeks, the tissue looks more or less like it did at week 0, plus the added weight gain of 6 weeks, which for mouse is actually quite a long time. So what happens to these cells? We can obviously do a molecular check. This is a bit of a complicated slide, but I think you can follow it. It's the same paradigm, but instead looking at histology in these animals, we now look at molecular markers that would denote the presence of brown or beige adipocytes. And I want to note here, these graphs are to scale, so you can actually compare amounts between the different tissues. In black, you have always the classical interscapular bud. You see in cold exposure, this is the upper right corner, there's a slight increase in UCP1, not a lot, that immediately drops back to original levels once the cold stimulus is removed. Interestingly, in the inguinal bud, you have a certain amount of UCP1, and this is increased quite dramatically. It's still 100 times less, or 50 times less than in the brown adipose tissue. And then it tapers off, and at 6 weeks, we're back at baseline. So the uncoupling protein, which we already heard about, and which is the functional marker of brown and also of beige adipocytes, seems to be quite tightly regulated in response to cold. If you look at markers that would be rather more cell-specific or mitochondria-specific, like COX7A1, you see that those actually stay high for 2 to 5 weeks after the removal of the cold exposure and then return back to baseline, suggesting that you have formation of activated cells, which then slowly are removed from the system. So now that we know this, we can actually follow these cells and ask what is the fate, and also not just what is the fate, what happens to these cells and how do they behave in response to various environmental paradigms. So for this, we built several new models. The first one I want to introduce here, which is a tracing model, where you drive a Cre-ER expression, so tamoxifen-induced Cre expression, under the control of the UCP1 promoter, which then removes a stop codon in front of an RFP. So if you give tamoxifen, all cells that are at that time UCP1 positive will be labeled with RFP. And you see here the paradigm. We give the tamoxifen during the cold, and then we follow these cells. And you can see here you label approximately 1% of all the cells in the inguinal adipose tissue. Around 30% of the brown adipose tissue are actually adipocytes. These numbers have been confirmed. It's actually, on a side note, quite important. Only 30 to 40% of the adipose tissue are actually adipocytes. The rest is something else. And quite interestingly, the number stays stable over time. It increases a little bit after 4 weeks in the inguinal, and then it goes back to the original levels. But it's more or less the same. It doesn't fit at all the high increase in mitochondrial markers. In epididymal white adipose tissue, I told you there's no browning, and this is very nicely confirmed by our model. You basically have 0 positive cells in this particular tissue. So this means that the cells actually are there and they remain within the tissue. So what happens to these cells? You can answer this by looking a bit closer at using a fluorescence readout. Same paradigm. These tracer mice are kept in cold while all UCP1 positive cells are labeled. And now we add a third mouse line on top where we express GFP, a membrane-bound GFP under the control of the UCP1 promoter. So if a cell is red, it was at the time of tamoxifen induction UCP1 positive. If a cell is green, it's at that current time UCP1 positive. So it's basically a surrogate. And this is when you have mice in cold. So cells are labeled in red because they are UCP1 positive cells and the Cre is excised. And you have the green membrane-bound GFP in these cells. If you now go for 6 weeks without cold stimulus, you remember that we lost the expression or lost the multilocular cells and also lost the gene expression. You see that these cells, which were formerly brown cells, are still present. However, these cells now look like they are white adipocytes. So these formerly brown adipocytes have turned into something that resembles, morphologically speaking, a white adipocyte. If you now re-stimulate with cold, which is shown here, this becomes quite interesting. You see that these red cells, most of them, actually revert back to forming brown cells or beige cells. In addition, there's some cells that weren't labeled in the first round. You see these just green cells that do not express any red color. So these must be newly formed beige cells. And in addition, some cells, like the one in the top left corner, remain unilocular. So these data indicate that you have a quite high plasticity within the adipose tissue. And I tried to summarize this here. We have probably precursors for brown adipocytes that generate brown adipocytes. We have precursors for bright and for white adipocytes, but if you form bright adipocytes, they have the ability to transition, at least on a morphological level, to something that would rather resemble a white adipocyte. Whether these are actual white adipocytes or whether this is a different type of bright beige adipocytes will remain to be seen. So you see already the plasticity is much higher than we expected then, and we have a certain possibility for interconversion. So what I want to show you next is some more data on possible mechanisms how to modulate this process. For this, I'll quickly introduce the concept of bone morphogenic proteins, which you might have heard before. So bone morphogenic proteins are morphogens that control stem cell fate, and it's quite well accepted in the field that mesenchymal progenitor cells can be programmed to become rather white pre-adipocytes with BMP4, which ultimately leads to the formation of a white adipocyte, while BMP7 is a driver of the brown adipocyte lineage. Now, in light of the data that I showed you before, that there seems to be a sort of plasticity within the adipose tissue, we were quite interested to look at whether BMPs, which are also expressed in the adult, also in the adult humans, would maybe be able to also change the cell fate, not just at the level of the precursor or even the stem cell, but maybe even at the level of the mature adipocyte. And for the sake of time, I can't talk so much about the precursors, although the data shows more or less the same, but I want to focus exclusively on the mature cell. So BMP4 is a secreted protein, which can be expressed from an adenovirus and is then secreted in a paracrine manner from the cells. And if you inject, for example, a virus into the normally brown adipose tissue depot, you can induce BMP4 expression. And at the right bottom corner, you see that this induces phosphosmart signaling, which indicates the presence of BMP4. Quite interestingly, and we are talking here about a fully differentiated brown adipose tissue, this already reduces the number of UCP1 positive cells, both on the protein as well as on the mRNA level. And I think it's even nicer if you look at this from a histological perspective. So please focus on the picture on the right, which shows you the brown adipose tissue in mice, either an H&E stain or a UCP1 stain at room temperature, the upper 4 pictures, and after cold exposure. You can see the Laxette control animals, which are always the top 2 figures, have a very dense brown adipose tissue, and this density is even increased if you stimulate mice with cold. In contrast, if you give BMP4 now for just a few days, you see these unilocular cells appearing, which would resemble rather whitish adipocytes. And even if you cold stimulate the mice, you can see that there is a strong shift towards formation of unilocular white-like adipocytes, suggesting that you have the capacity not only to convert if you remove a stimulus, but also through signaling mechanisms, in this case bone morphogenic proteins, can actually change the cell type from at least if you look at a morphological perspective. Quite interestingly, and this is now a bit of human data, BMP4 can be measured in the blood. It's probably a rather paracrine factor, so we have to be very careful with putting too much emphasis on the blood data here. But if you look at BMP4 levels in a cohort of lean, obese, and diabetic people, you can see that the more BMP4 is present, the higher the chance for a person to be diabetic. BMP4 is also reduced, expression is reduced in human brown adipose tissue, and the levels are correlated with BMI, the bottom left graph, adipocyte diameter, which would fit with our observation. BMP4 promotes large unilocular cell formation, and also the insulin infusion rate within a clamp. So it's very well possible that within humans there are certain differences in BMP4 levels for whatever reasons, and that these levels determine the ratio of brown and white fat cells by reprogramming cell fate, either from the precursor or maybe even at the level of the mature cell. And this could also contribute to a certain extent to the obese phenotype and also the metabolic changes. So I wanted to put this here into context. I didn't show you much about the precursors, but BMP4 is able to reprogram those as well. So you not only have the capacity that bright and brown cells can form, morphologically speaking, white adipocytes, but that you can trigger this conversion both at the level of the precursor with, in this case, morphogens, but possibly other factors. So for the last 5 minutes of my talk, I want to really focus on another aspect that also elucidates or illustrates a little bit this paradigm of plasticity in the adipose tissue. And this will introduce a bit the concept of epigenetics in controlling this plasticity. This study started with the analysis of a human cohort in which we looked at PET-CT scans from about 14,000 patients from the greater area of Zurich. And in the upper left corner, you can see how a person with active butt looks, while a person with inactive butt has basically no brown fat. What was already quite interesting, that especially in the colder months, when brown fat is probably more activated due to outside temperature changes, we found that people that had active butt tended to be a tad lighter. But what was even more striking was, which brought us to study the concept of epigenetics a bit more, was the fact that if you correlated brown fat activity with the projected months of conception, in this case, we found a strong bias towards certain months. So this is shown in the right corner, and you can either look at slightly activated, that's on the top, or more strongly activated people. And you see that there's a really high level of fully activated humans with lots of brown fat, which were conceived in the colder months of the year. So we were quite interested to look at this a bit closer, and obviously you can't follow humans over 20 years based on whether they were born in winter or summer. So we again went and utilized the mouse model, and you can see here the different groups that we employed, where mice were kept at either room temperature or at 8 degrees, and then were mated. And in this case, both the father and the mother was kept at room temperature or cold and then mated. And here on the right side, you can see UCP1 expression as well as the quantification. And as you might see from the quantification graphs, if you mother and father in mice in the mouse model were exposed to cold before conception or during conception or during the first few days of gestation, the first 2 weeks, all had the offspring of these mice had much higher UCP1 levels in the brown adipose tissue as in the basal state, that's the upper panel, as well as the activated state. So this is quite interesting as it actually reproduces a bit the correlation which we saw in humans. So we were a bit more interested to look at this a bit further, whether the father is at fault here or the mother. In mice, you can obviously test this quite well. So I can keep this quite short. I think in the upper graph, you can see here control offspring, maternal cold exposed offspring, and paternal cold exposed offspring. And as usual, it's the father's fault. So if the father was exposed to cold, the offspring had quite a significant increase in brown adipose tissue UCP1 levels. And here again, you can see the density, which is a very good indication of how active brown adipose tissue is. And you can see here on the bottom right side, these are cold exposed offspring or offspring from cold exposed fathers, while on the left side are the control offspring. So the brown fat seems to be much more activated. So since we obviously want to talk about metabolism here, we were interested, does this actually really translate also into altered whole body functionality? And you can see here, yes, it does. You can even with a heat camera see that PCE offspring are a bit warmer in the area of brown fat. But what is even more striking, you can measure oxygen consumption of these mice. So on the bottom left is an oxygen consumption graph in which you measure how much oxygen a mouse consumes. And you can keep them at room temperature and then induce cold and then adapt them back to warm. And you can already see even at basal, in a basal state, the mice from cold exposed fathers had a slightly higher energy expenditure. If you calculated all, I think in total we looked at 40 or 50 mice, this is even significant. It's about 5 to 10%, which energy wise is a huge difference. But it became imminently more clear when you cold induced these mice that their energy expenditure would go up. And on the right side, you can see the quantification of this graph. So how does this translate if we talk about obesity? And I wanted to show this since obviously it's an obesity seminar. It's a small change. We're talking, as I said, about 5 to 10% differences in energy expenditure, but this really translates into differences in weight gain and a reduced fat mass if your father was cold exposed. Actually, it seems like these mice compensate to a part for their enhanced energy expenditure by eating more. On the very right, you can see the food intake, which is higher if you are derived from a cold exposed father. And obviously, you also see a change in energy expenditure and temperature. And if you take this even one step further, you will see that this changes in UCP1 expression, which is shown on the left side on an obese model, also translate into altered glucose tolerance. So here you see a glucose tolerance test where mice were given glucose. It's an insulin tolerance test where mice were given insulin. And you can see that mice derived from cold exposed fathers are not only lighter, they are also metabolically much healthier than their counterparts, which were born from a regular system. So if I summarize all that I've showed you, we have a high plasticity in the adipose tissue, not only at the level of the precursor, which you've also seen from Camilla's work, but also at the level of the mature cell, which can be driven, as I showed you, by BMP4, but there's many other factors that can regulate this process, but also by environmental stimuli, which control, in this case, the DNA methylation patterns and might then give access to DNA regions and change either the cell fate or even reprogram a cell on the mature level. With this, I'd like to close, and thank you very much for your attention. I would like to point out the people who actually did the work, which were Salvatore Modica and Wenfei Sun and Matthias Rosenwald. thank you very much for your attention, and I think this closes the presentation part, and we are very happy to now answer any questions you might have regarding what we just told you about. Thank you very much.
Fantastic. Thanks so much, Christian and Camilla, for the fantastic presentations. And with that, we're going to move right into the Q&A. We have had a number of great questions come in already, but as a reminder, if you'd like to submit a question, you can use the Ask a Question panel to the right of the slide viewer. And so let's jump right in. First question today is for Christian. So you talked about the ability of brown and white adipocytes to interconvert, but are there actually more types of adipocytes besides beige and white ones?
This is actually an excellent question, and I guess I would say yes, there probably are. The thing is that it will require further analysis and single cell analysis of mature adipocyte fractions to actually define what other types exist and if there are really other types or whether they just resemble certain states of an adipocyte. I cannot give you a definite answer at the moment, but from everything I've seen, I believe that there's probably various other types that we have not yet identified.
Excellent. Thanks, Christian. Next question is for Camilla. It's about applications of this research. Camilla, do you think it's possible to utilize these mechanisms to treat obesity?
So it's very clear that there's an effect in mice on obesity if you activate the thermogenesis. In humans, it's a little bit more complicated because you usually don't put a human into 4-degree room for 3 weeks. And also the amount of brown adipose tissue in humans are also smaller. However, there is clearly an effect on human metabolism if you activate thermogenesis. So, for example, we have seen from intervention studies either using cooling or pharmacological stimulation of beta-adrenergic signaling that this increases energy expenditure and it also improves insulin sensitivity. So there's definitely potential. I think also we need to look into more the roles of secreted factors, cytokines, how they affect metabolism. They can interact with different organs. There is also an example of a pathway where you have the gut peptide secretin that actually signals via activating brown fat and then mediates a satiety signal of the brain. So, I mean, in the long-term perspective, I'm quite certain that we will see an effect on how brown fat regulates body weight.
Excellent. Great answer. And here's I guess a similar question, but Christian, I'll direct this one to you. Is the amount of BAT in adult humans not too small to have an impact on metabolism?
This is actually a question. If you think about it, how do we gain weight, right? Nobody gains 10 kilos in a few weeks. You gain 2 to 3 kilos a year. That basically boils down to 50 kilocalories a day, one cookie or something like that. And that's actually not a lot. And that could easily be utilize this little bit of energy by brown fat, right? So we are talking more about a preventive angle. On the other hand, if you want to really lose fat, it would also take a while. And you must never compensate by overeating this extra amount of food. So I think we'll have to see. Probably the biggest advantage will be a combination of weight loss drugs that induce satiety, maybe in addition to drugs that activate brown fat to enhance energy expenditure.
Excellent. All right, here's a good question that came in from Karen. She's asked, why is it that hormonal activation of BAT is so much stronger than cold stimulus? So Camilla, this goes back to the start of your presentation, so I'll direct it to you.
So I assume that this is concerning the picture that we see in the pheochromocytoma case. So this is not a normal situation. So this is a disease situation where you have a chronic, highly elevated levels of norepinephrine pumping out from the adrenal gland. So this explains why it's so powerful. in the normal situation, it's the activation goes mainly through the sympathetic activation, so it's directly targeted to the brown adipocytes. But when that is said, it's also there are also healthy interventions that shows that, I mean, for example, if you go winter swimming, putting yourself into cold water, this will also give you a shock and will also give you like a hormonal onset of adrenergic activation. So then you will have a more targeting of the vascularized adipose tissue, I would assume.
Excellent. All right, great answer. Here's an interesting question. We have had a couple questions already come in about exercise. Can exercise influence interconversion of white adipocytes to brown ones? Or also, what's the relationship between exercise and BAT activity? Christian, maybe you can lead this one.
Yeah, so I think this is not yet fully resolved. There is a very interesting study from the Spiegelman lab suggesting that irisin is a myokine that induces browning. There has been more recent work that just came out recently suggesting that whether it induces interconversion or de novo formation from specific precursors is I think not clear yet. On the other hand, we have to be careful because exercise might also influence metabolism per se, and thereby change how the fat is composed and indirectly change interconversion. I don't think it's fully resolved. There is some evidence that there might be an induction of bright or beige adipocytes in relation to exercise, but the exact mechanism is also difficult. Yeah. Excellent.
Another question here, Camilla, a bit of a basic question here, but can you just clarify what exactly are bradykines and what are some examples of them?
So bradykines are simply just proteins, or it could also be lipids, have been called bradykines, but let's say factors that are secreted from brown adipocytes and have a role of activating either acting in a paracrine fashion or endocrine or autocrine. So it's a wide, I would say, wide concept of factors coming out from brown adipocytes and signaling to other cells. So, for example, we have identified a bradykine called EPDR1 that mediates thermogenic determination. And then there are other examples would be FGF21, secreted from, actually, we haven't really seen it much secreted from human brown adipocytes, but it's well established. It comes from in mice. It's secreted, and in humans, it's probably coming more from the liver, it seems. So there are a handful of factors that are identified. IL-6 has also been mentioned as a bradykine, and is of course also a myokine. So it's more of a factor that can come from several different cell types.
All right, perfect. Question here for Christian. Why was it that cold-exposed fathers contributed to BAT activity in offspring more so than the mothers?
So we don't fully know. What we know is that the cold seems to reprogram the epigenetic memory of the sperm, and this leads to a change in both brown adipose tissue innervations. We're really talking rather about a developmental phenotype. Yeah. So the epigenetic reprogramming in sperm induces a reprogramming and development that induces the innervation of the brown adipose tissue and the vascularization, thereby leading to higher activatability, I would call it. On the other hand, it seems to also shift the precursor balance, which is a bit more what Camilla talked about, but probably some of the pathways she's working on are involved there. So you see higher formation of brown cells from adipocyte precursors, suggesting that this insult or this change in physiology elicits a change in the precursor pool or reprograms the precursor pool. We don't really know this. This is probably due to the fact that sperm is much more easily reprogrammable than an egg. So this is why probably that is a paternal transmission rather than a maternal transmission. We don't know exactly how the cold stimulus is sensed, whether this is also coming from the beta-adrenergic system or whether there are other temperature sensors involved at the testes that could explain this result. But yeah, so especially the epigenetic memory as a paternal driver is what we've seen. That's why it's more paternal and exclusively paternal, not just more. It's exclusively paternal and non-maternal.
Excellent. Great answer. Camilla, I've got a question for you. What is it about perirenal brown fat that functionally distinguishes it from other brown fat depots?
So I don't think it's really known what the differences are besides the location. I mean, when we looked at the tissue sections, it looks very similar to what we see in the supraclavicular region, and also when we isolate the pre-adipocytes and differentiate them, they're also very similar. So we haven't seen a very big difference actually, but we haven't compared them globally to each other either. But we are in the process of doing that, so I think we will know more in some time.
Excellent. Another question here. So more broadly, maybe than exercise, but does weight gain or loss affect brown adipose tissue in humans? Christian, I'll direct this one to you first.
This is actually really an excellent question. We really don't know yet what the chicken and what the egg is, right? I mean, we know that people with less weight or less fat mass tend to have more brown adipose tissue and vice versa. So we can argue that these people who have more brown fat tend to gain less weight. We can also argue that somehow weight loss induces brown adipose tissue formation, maybe to compensate for the missing insulation. So this is something we will really need to figure out in the future, especially for humans, if the brown adipose tissue correlation to obesity is due to the fact that the weight loss drives adipose tissue browning, or whether the amount of accessible brown adipose tissue induces the ability to lose weight. Now, I'm sorry, I can't give you a more clear answer, especially not for humans. In mice, it really seems to be that browning drives weight loss, and not the other way around. But in humans, it's totally unclear. So weight loss induces browning, yes, but we don't know whether it's causative in the weight loss or whether it's just happening because we are losing weight, right? I could go into it with a bit more detail. It's a complicated question. One argument is that if you lose weight, you have a lot of excess lipolysis, and it could very well be that the brown fat is also necessary to get rid of the excess lipids. So in that case, it would make sense that the brown fat is a secondary event to weight loss and not just to drive weight loss. But it's a thought, right? It's not proven, and we have to work more on that.
Well, that's a fantastic answer, and I think we'll end on that high note. So thanks so much, Camilla and Christian, for all the fantastic insights today, both in your presentations as well as the Q&A session.
Thank you very much. It was great. I've done it for the first time, but it was a fun experience. Thanks to the audience for listening to us, and I hope it was of some interest to you.
I also like to thank everyone for listening, and it was an interesting discussion as well. So thank you very much for staying in for that.
So in closing, thanks again for taking part in this Inside Scientific and American Physiological Society webinar, and we look forward to having you with us again soon. Have a wonderful day, everyone.